Welcome, everybody, to our journal club. We are not covering a Pavlovian paper this week, but we are covering, of course, a lifespan and aging-related paper as per usual. So I'm Oliver Medvedik, along with everybody else that's joining us, our heroes. And I'm just going to quickly say that there's a special message that we are having our end-of-year fundraiser, Giving Tuesday, which is, I believe, today. So Steve might put up a link at some point, but please check out our website for that. And I believe there's some voting taking place for favorite projects. But today is Giving Tuesday, so if you would like to open your heart and your purse strings and visit our site and donate, please do. I encourage you, of course. So without much further ado, let's share a screen and see what paper we're doing. Oh, this is the paper. Okay. It's the Shinyapina et al. Paper with our friends who've presented at our conference, Steve Horvath and Vadim Gladyshev, along with Leonid Peshkin, who I believe has some of his work on our website. And the title of this paper is Rapamycin Treatment During Development Extends Lifespan and Health Span of Male Mice in Daphnia magna. If this sounds a little bit familiar, it's because we covered a paper last month that dealt with health, basically improvement of certain health characteristics of mice when they were treated transiently for a small period of time relative to their lifespan with rapamycin. In this paper, they're extending this to not just mice, but Daphnia magna, which is a small metazoan multicellular organism, an aquatic organism. I believe it's also called a water flea colloquially, and demonstrating that the health span of, or lifespan actually, of Daphnia and the health span and lifespan of mice is extended. So basically, two widely divergent organisms on the evolutionary spectrum are affected by transient rapamycin treatment. So, okay, and one thing to point out is that they use, in this case, genetically diverse UMHE3 mice, which I believe come from 4 different mouse backgrounds that have been mated together, so a little bit more genetically heterogeneous than your typical C57 black 6 mice, for example. And let's see what they do. So I'm gonna scroll down a bit here. So just as a reminder, everybody, rapamycin is a drug that's basically was isolated in a soil-dwelling microorganism on an island. I love this story because a lot of these small molecule drugs have been isolated from microbes that have been found in the soil. Antifungicidal like nystatin found in soil samples in New York State. That's why it's called nystatin. And rapamycin was isolated from a microorganism found on the island of Rapa Nui, which I believe it's in the Easter Islands, somewhere around there. So way out there. And it's used as basically, well, as a kind of it affects the cell cycle of a lot of cells, but primarily T cells. So it's used as an immunosuppressant drug, and it might have anti-cancer effects. So it has a lot of different wide-ranging effects, but it was initially developed as an immunosuppressant drugs to prevent organ rejection. So yeah, a lot of cool history in finding these small molecules. People basically mucking around in mud around the world and identifying these things. I don't know if they do that as much as yeah, Steve.
He did notice it did work. We've already got a question which is related to what you're saying. Rapa Nui, I understand, is the name of the island. We would probably all call it Easter Island, but in the native language, I understand it's called Rapa Nui, and I'm going to go out on a limb and say that the language could be based on ancient Phoenician, but I couldn't tell you what specific language group it is on that island. So there you go. We've got a question from Facebook as well. We've got Madonna Chakota. Sorry if I've actually butchered that name. On Facebook has asked a question. They want to ask if this drug could be used for immune suppression for an autoimmune disease instead of a TNF blocker. Any thoughts on that one?
If it could be used as for autoimmune diseases, well,
yeah, don't they already do that now?
Good question. I don't know. A lot of this stuff comes down to dosage. I don't know if it is. I am not a medical doctor, so I don't know if it would be prescribed or if it's actually used off-label. Sometimes doctors do that. I don't know. Maybe one of our guests knows more details as to how it's medically applied in other indications.
I don't know about autoimmunity, but the traditional on-label indication for rapamycin is an immune suppressor for like kidney drainage.
Yeah, and it's worth noting at this point, because what happens is a lot of people get the wrong end of the stick with rapamycin, and they think it's only used for that, which it does obviously suppress the immune system, but the kinds of dosages that people are interested in using for like longevity purposes are minute in comparison to the actual use that they for immune suppression, because they use it for organ rejection in the healthcare system today, but the dosages are much higher than typically seen in like longevity based experiments and research, right, Oliver? I mean, it's tiny in comparison. So because people say, oh no, it's going to shut all your organs down and your immune system. No, not really. Dosage is a really important thing.
Yeah, it's an important thing, and they show that with they mentioned briefly that I don't recall off the top of my head. It's in the paper, of course. The dosage they used for the mice here, they fed it in this methacrylate carrier. They mentioned that in the paper. Hi, John. Yes, hand up. You're muted.
I was unmuted. Oh, there we go. All right. There is a select forum of rapamycin users, and as somebody who's an intermittent rapamycin user, I'm part of that forum. I think the issue isn't necessarily the dosage, but the frequency of the dosage. Because it basically inhibits IL-2, that's where the immunity aspect comes in. But it's because we're inhibiting mTOR and the IL-2 response of mTOR. But it's not so much the dosage as the frequency.
Okay. Well, there you have it, folks. That is, so we have some statements to how it's used from experience. I have not personally taken it, so I don't know. So, okay, so let's go back to journal article. So back to rapamycin. So this is their protocol. Dams with newborns. They mention how many mice they. Does John have it? Oh, I guess that was probably from before. Let's see how many. So they used quite a bit of mice. They had a cohort of 130 newborn, UMHE3. I don't know how you pronounce that, just that, or het mice, 3 mice for a lifespan experiment, and a separate cohort of 40 mice for Healthspan analyses. And speaking of dosage, they fed them chow containing 42 parts per million of this thing called gait, encapsulated rapamycin, which I mentioned earlier is this methacrylate stuff, which is basically a carrier, an encapsulating material, or they just fed them the encapsulating material as a control. And this proceeded until the first, basically after they were born, and for first 45 days of age. So, and then they were transferred to a regular chow. So that was their protocol. So basically the first 45 days of their life. So let's scroll down here and see basically what are some of the effects of this protocol. And after doing this, they do a whole bunch of analyses. So they look at, so a lot of these analyses were done at young mice, middle-aged mice, and old mice. And these are the time points: 2 months old, and then 20 to 22 months old and 28 to 29 months old. And they do frailty index, gait speed, and then they do a whole bunch of genomic stuff. They look at DNA methylation, they look at genome expression, so on and so forth. And they also look at various physiologic parameters, such as glucose regulation and insulin levels. So one thing they noted is that these mice, after being treated, and how long is this? So they do tend to be smaller. So it's kind of not too much of a surprise, I think. Or maybe it is. I don't think so, because it does affect, cell cycle regulation. So the mice are smaller, representative age of 2-month-old mice subjected. So body weight change subject to rapamycin. Let's see. Don't know exactly what date they looked at them here, but they tend to be smaller. So body length millimeters control rapamycin. The weight tends to be smaller in mice, both male and female. So they have male and female mice. So the weight differences, and basically it's correlated with this overall size of the mice. They're smaller. That's the same between both male and female. We'll see later that a lot of the positive aspects when it comes to lifespan tend to apply primarily or only to the male mice rather than the female mice. But the effect on weight and same thing, and overall size of the mouse is for both. So what else are they looking at here? So one thing that they did note here is that, let's see, figure G, figure E, they want to ensure that this weight loss isn't really because the mice are eating less. So what they do is they have these mice that are basically, weaning off of the mother mouse, right? And then they remove the pups after 6 hours, and then they weigh them, and then they return them, and then the mice feed, right? And then they basically see what the difference is between body weight gain, and that presumably is the amount of milk they drank. Then they induce milk secretion in the mother mice, and they analyze protein concentrations to make sure that the mother mice aren't just that there's a sufficient amount of milk proteins there, and it's not because they're not getting enough proteins in their diet. And the concentrations of protein in the milk using these assays, and the percentage of body weight gain, which is attributable in a very short period of time using this assay, they conclude that it's not because there's like a loss of appetite in these mice, or they're, and that might be contributing to the overall loss of mass or weight. So it's not that they're calorically deprived per se. Let's put it that way. So I don't know if anybody has a comment on that, but I thought that was good that they did a little bit of a deep dive there. Maybe a reviewer asked that. Perhaps they thought of that themselves, but they dove a little deeper to make sure that that's not accounting for that loss of mass and that the size difference. What else did they do here? One thing that they noted though also is that that the litter size per se did not change. So this is pups after they basically became sexually active or sexually mature, but the days to birth was a little bit longer. So it took them longer to basically achieve pregnancy. It was a very short period of time. I think it was about average of 4.6 days longer. And that was only evident between litter 1 and litter 2, but not, litter 3. So they were concluding, what they concluded was that, that the smaller stature and size of these mice, was due to some other effects of rapamycin, wasn't due to the fact that they were getting less nutrients from the moms, and that the age of reproduction was slightly shifted forward, which, correlates sort of with the evolutionary, theories of aging out there, where you have a postponement of age of reproduction. But how rapamycin is potentially doing that, not quite certain here. But that is noted in these assays, days to birth assays that they performed, and that's figure 1 on the effects of rapamycin so far on overall mass of these mice, and a delay in sexual maturity. Although, when I should say delay of giving birth to litters, right? I'm not sure if they looked. I don't think they looked at like hormonal levels or other types of attributes of a sexually mature mouse, unless it's in the supplemental. So I'm going to stop sharing here. Anything intriguing about this figure that somebody would like to comment? We haven't gotten to the aging stuff yet. It's all just fundamental stuff. Okay. All right, so let's move on to figure 2. And figure 2 is the big one. We've always got people asking, do they actually live longer? Yes, they do. All right, let's move on. No, let's not. Here's figure 2. So as I mentioned, this effect is seen in male mice. It's not seen in female mice. Obviously, if you average them, then you get an average here, extension of lifespan, both male and female. But of course, if you parse it further and you say, well, is it really male or female? The effect is primarily from, so this right here, the average lifespan, or I think this is the average lifespan, the p-value. It's not significant. It doesn't look like the maximal is extended. Here you can clearly see there's a maximal and average lifespan extension happening with the rapamycin treated mice that are male, right? So these are the ones that treated during the first 45 days. So there is a significant extension. So the survival curve is improved. So they're, despite their smaller mass and, slight delay in age of reproduction, they are living longer. So these are the UMHE3 mice that are genetically heterogeneous, at least to the extent of coming from 4 different mouse backgrounds, which I can't recall which 4 mouse backgrounds there are. But anybody wish to comment on this figure?
I kind of love this figure. I know it's really simple, but coming into this paper, there's in aging, I think it's fairly well known that if you're in many animals and people, being bigger is generally not so great for your longevity. Mm-hmm. Although that's not true between species. Bigger species typically live longer, but within species is not so great. There's a lot of possible reasons that might be true, and it's kind of crazy that they can recapitulate at least some of this effect with a single small molecule. Like, no one's going to take rapamycin therapeutically as a kid. That's a non-starter. So when I look at this paper, it's like more mechanistic. Mm-hmm. Like, what's the deal with changes in body mass or the association with body mass and height and lifespan? And this is like one of the cleanest experiments I can imagine showing that, oh, all right, you're smaller and now you're going to live longer.
Yeah, and we'll see later that there's a lot of different effects happening between like male and female, and there seem to be a lot of kind of negative effects with some of the things happening when they look at the transcriptome profile in the female mice, although that doesn't seem to be affecting their lifespan. So it's like, I don't know what's happening there, to kind of some sort of counterbalancing happening with the female mice, but yeah, this mass difference that spills over into the water daphne as well. So just letting people know that that's coming up in figure 5, I believe. Yeah. Yes, John.
Well, if you inhibit mTOR, obviously you're going to get less growth. I think the point about like turtles is the turtles that live a lot longer are actually bigger. So I don't think the size of the entity is such an issue. My issue with this paper is an issue with generally how people look at rapamycin, which is that the arguments about autophagy, and I found it interesting, the word autophagy does not appear in the paper, but appears actually in one of the references, but not the paper. I just searched the paper to see if it was there. And rapamycin is, as I understand it, to be thought to be particularly useful in encouraging autophagy through its inhibition of mTOR. That's a cycling process. That's something which the question is, a lot of the people who take it weekly. My personal view is that's too frequent. And I haven't taken it the second time yet, and I probably will take it 3 monthly. Now, this is a paper based on taking it basically in all the food. And given that it's like a lot of things have multiple effects, and encouraging autophagy from time to time is a good idea. And if it improves the quality of the mitochondria, that's obviously a good thing. But you don't have to do that every minute of the day, every time you eat food. And it's like fasting. Fasting is a good thing, but you fast forever, you die. And so I do think that there is a problem inherently in the design of the research, that it sort of says eat rapamycin all the time. And yes, that's useful, but people are smaller. The mice are smaller. But I would say that's an inherent flaw in design of the paper.
Interesting. John Ferber.
Yes, can you hear me okay?
Yep, loud and clear.
Okay, I was wondering how they picked the dosage and whether future studies might look at a variety of doses and see different results.
Yeah, that's a good question. I don't know how they picked the dosage, but I do know that they later in the paper they do talk about that to some degree, and they mention that they used that they saw a lot of effects, a lot of the similar effects with water daphnia using very, very small dosages, and then seeing negative effects with higher dosages, and then commenting that some of the future experiments that they'd like to do with the mice is to lower the dosages. But I don't know how this came about as a starting dosage. It's probably based on a prior paper that looked at rapamycin, but I don't know how this particular one was calculated for the first 45 days. But they do mention they'd like to do a lower dosage regimen in the future and see if they would get similar effects.
I'm wondering whether maybe some of the scientists in our audience might want to do some dose ranging studies. Yeah. Well, if they're not, then I'm pretty sure the Gladys Chef and the lab and colleagues will. So the discussion is actually, it's a great discussion. I love discussion. I'm not going to unfortunately today have time to unpack the discussion, but I'm just going to go to the next figure. I'm just going to mention right now, so if people want to read the paper, I think the folks who wrote this paper, they pack in a lot into the discussion as to really kind of twisting and turning and trying to figure out like what can possibly account for the differences between the male and the female responses, and they give a lot of a lot of potentialities and hypotheses they throw out there. Do take a look at that. Maybe we'll if we have time, we'll get to some of the points they raise. Okay, so now they start looking. So yeah, lifespan is extended in male mice. So great. So this is good, right? They put usually you bury the lead, right? And it's like boom, they put that right in the front. And there's the lifespan. They're living a lot longer. Now let's try to see why they're living longer versus let's take a look at all of the stuff that that is slightly pointing in the direction of a potential lifespan extension, and then we get to the last figure and it's like, oh, but it doesn't extend. So we know it extends, but why, right? So they look at rapamycin treatment during development extends health span of mice, right? So there's one hint right there that the health span is extended, and they do this frailty index score, which they mentioned. They look at a whole bunch of different data points, and then it's an aggregate score. Here they're looking at also run times. There's a little iPhone app that I guess with a little stop sign that tells you how fast the mouse you let it go by the tail and how quickly does it plow into the back wall, I guess. 2.51 seconds. All right. So time to finish. And then they give it a frailty index score. And they do mention what it's a composite of. It's like 30 different parameters. And I believe they're all physiologic parameters. So the frailty index score is lower, which is better, p-value of 0.037. And is that a composite of male and female? Or is it, let's see, I think it is, of both of male and female subjected for, okay. So that's both male and female. But obviously we saw that the lifespan was only in males. Frailty index scores of females is also lower, but you notice that in later ages, it basically starts to converge onto the frailty index score. And this is something that was noted later in the expression profiles of looking at of various genes. They noted positive correlation with genes that are upregulated, that correlate with longer lifespan in females as well as males, but it tends to be, and also in some cases for the males as well, but was not as persistent in the females, right? So we kind of see that with the frailty index when you separate the males and females, that it's improved early-ish on, like later. I think this is probably middle age to the end, but this tends to persist relative to the controls. And then, of course, based on these frailty index scores, you get this predicted survival, right, which is used as a proxy, I believe, for lifespan. And that, of course, is improved, as we saw with the actual lifespan analyses. Time to finish this little race here. I believe it was more evident in males, negligible for females. So again, I think more of an indication, this is later in life, that the frailty index is more telling for the males than the females. They do mention something here, that this figure D, which is this little app that they show here, I think they're pitching it in this paper as a really kind of neat and quick way to do a fast frailty index sort of assay that basically gives you very similar results, and that it is a much easier way for researchers to basically get such a measurement. So that was mentioned in the paper. I don't have the link to this app, and I'm not sure. Maybe it's in the author's website, but it's there. So if you're doing mouse analyses and looking at frailty indices, this app-based approach that measures speed might be the way to do it. Might be a faster way to do it. Next, they look at glucose and insulin tolerance tests. I believe these are kind of classic tests to look at how quickly glucose basically is taken out of the bloodstream and how rapidly insulin acts. So I believe the glucose tolerance tests are on F and G, and insulin on H and I. You have a much better effect in males than in females. So with these, so P 0.22, 0.45, so not significant, but you do have a significant improvement. And let's see, how old are these? Are these the middle-aged mice? Must say it's somewhere in the depth of the paper if this is which age group that this mouse is in. Probably middle age. Let's see. Here it is. Determined using a photo finish app. All right. We further developed and applied a new test to assess mobility fitness in mice that does not require specific equipment and can be conducted by using a cell phone and a common app. So there you go. You can use your iPhone to measure how fast your hamster runs, and DIY rapamycin experiment. I don't think there's been a rapamycin hamster paper, so just throwing that out there. Okay, let's see. John Ferber, you have a question?
Yes, I just wanted to mention that the speed of walking or speed walking is used as a human biomarker and is easy enough for any of us who might want to be testing ourselves, maybe weekly or monthly. You walk for 6 minutes as fast as you can without running or jogging, no trotting, and you just make a note of how many meters or how many feet you go in 6 minutes. It's called the 6-minute walk test, and it's a useful human biomarker.
Very good. Yeah, there's a number of these fairly simple tests. There's one, I believe, like sitting up from a chair, and other types of tests, which and they actually are despite their simplicity, they're actually pretty powerful predictor. Assuming, of course, you control for any confounding variables, like somebody's missing a leg, for example.
Well, one nice thing about the 6-minute walk test is it's numerical, and you can look at a graph, whereas standing up from a chair is just, can you do it or not, which is a measure of frailty. But the 6-minute walk test, you can actually gather statistics for a long period of time.
Okay, well, all right, so that it's even more powerful. Okay, so I'm just looking at what age they use the tolerance test. Treated males of older ages. Okay, I'm going to just assume that it's middle-aged and up for these glucose tolerance tests. And what do they do here? J and K. Oh, that's just basically area under the curve. It's just different way of representing the data that you saw in figure F, G, H, and I down here, just to show you that it is a male effect, not a female effect, with the glucose tolerance tests. Okay, so now we got a whole bunch of kind of genomic data here, and we have experts that can try to tease apart or notice something that's kind of really popping out. Please do chime in, because it's really kind of difficult to figure out what and the authors are talk about this in their discussion, because there are a lot of differences here. So figure A. So they're looking at figure A. Let's kind of unpack this. So they look at DNA methylation status. So that's no doubt done at the Horvath lab with a pioneer of methylation clocks of aging. And again, a lot of these profiles you'll see when we bounce back there, they tend to kind of are improved throughout kind of the lifespan of the male mice, but not so much the female mice. So they looked at first in A, differentially expressed genes in the liver and kidney of young, middle-aged, and old animals treated with L-rapamycin is not the Spanish version of rapamycin. It is early life rapamycin. That's their term for it. So treated with early life rapamycin and compared to controls that haven't. So these are basically, and what is on the, number differentially expressed genes. So that kind of peaks at something beyond 60. And I don't know what those, probably in the supplemental, what those differentially expressed genes are. So the number, so basically that's just the difference between a control animal and one that's been dosed after being dosed with 45 days with rapamycin. And it's broken down here in figure A, sex, female, male, and then combined both. So what you can see here, that in the liver, that really kind of in the young mouse, it skyrockets, right? And, you can kind of ignore the both in this case, right? If you've got both the male and female, then I don't know, it's not really telling too much to have kind of both combined here. But you notice that it's a much more pronounced effect, that just overall different genes being turned on in the male mouse. And that gets attenuated for both male and female in middle-aged and old. Kidney, it's the same type of attenuation, but I'm not really sure, why per se, they would look at liver and kidney. So I'm not, versus other organs like pancreas or, I don't know, parts of the small intestine. But they're looking at liver and kidney. I couldn't really find the exact reason there. But I think their take-home message is that a lot of these effects are pronounced early on in young, and then basically the expression profile drops as the mice get old. So that's something that they kind of scratch their heads on later in the discussion as to what can account for this transient effect that has a long-term effect. And, clearly something to consider is changes in the epigenome potentially, or other effects. They also mentioned that it could be an attenuation. I'm just jumping ahead to what I remember reading in the discussion. Another possibility is I don't think we read this paper, but we do know that the rate of somatic mutations, or the somatic mutation load, right, of the genome correlates with the lifespan of organisms across species. So one possibility they mentioned is that there might be some attenuation of somatic mutation load early on, which, that's a ripe area for investigation with this transient rapamycin treatment. And there's other things that they postulate. Okay, so you have this transient effect, association between gene expression changes induced by early lifespan rapamycin, B, measured in young, middle-aged, so same colors but different measurements here, in young, which is red, middle-aged green, and old blue animals, and signatures of aging and lifespan extension in males, top plot, and females, bottom plot. And these intervention signatures, they compare that to gene signatures of individual interventions, of different individual interventions. So they compare the treatment of the rapamycin transiently treated mice with other interventions. And I don't know, I guess this is from a database because they didn't, and I'm assuming that it's not probably also from the UH-MET3 mice, it's probably from other mice. So calorie restriction CR, growth hormone GH deficiency, growth hormone deficiency, and rapamycin common gene expression. They also look at, I believe, let's see. And signatures associated with the effect of interventions on maximum or medium lifespan, max and medium. Okay. So let's see if we can find from this plot here. So NES is, what is that? Normalized enrichment score. Don't exactly know how to normalize it. So males on top, females on bottom. It's kind of just glancing at this, you could see a difference, right? I mean, so you could see, so young is red, green is middle-aged, blue is old. So they're looking at, so anything higher, I believe, than this baseline here is I think is a higher enrichment, or is a difference in gene expression. It's a higher and different gene expression than the intervention they're comparing it to. And one or something within these boundaries would be similar. If anybody wants to chime in on the interpretation on this, I would be grateful, but that is my interpretation.
I'll just chime in that it's kind of really challenging to interpret RNA sequencing from these whole organ experiments, because it looks like for their methods they take plugs of liver and kidney and they do sequencing on the whole thing. Liver and kidney both have a bunch of cell types, and if the composition of your organ changes due to your treatment, that's going to change your results. You also don't know, that could entirely mask the effects they're looking for. Usually the stuff gets a lot more interpretable if you sort out specific cell types for analysis, or if you do some kind of computational analysis to sort of pseudo sort out the cell types. I'm not really sure how much you can read into this when it's just like the bulk tissue, unfortunately.
Yeah. Let's see here. So on the right, on the left is the interventions. On the right are let's see. These are, I think they're comparing it to so they have a brain, liver, which is, I believe, from a mouse. Then it's human, rat, and mouse. And is that all genes? So I have to come back to that. I apologize, because I have to figure out what, which tissues this is all coming from. Yeah. Be that as it may, when they look at the liver, so now they look at the DNA methylation age. So this is the kind of also one of the other intriguing things, that the DNA methylation age, when they look at the young mice, it correlates with a even younger DNA methylation status, but that's not, and I believe this is in males, C, not females. And young and treated animals. Or is this both male and female? It's kind of important to know if it's male or female. So there seems to be a positive correlation with DNA methylation age, but only, and they mentioned that in the discussion, only at the young stage, but not middle-aged and old. And I don't know off the top of my head which CPG islands they're looking at here. But they note it's a transient effect. D, E, F are basically looking at transcriptome profiles in both males and females, using these graphs here, which basically the color is basically indicative of the increase in fold enrichment. So red is going up and blue is going down. And then the size of these dots is the negative log of the p-value. So if they're bigger and the p-value is smaller, then that means the dot's bigger. So basically, the bigger the dot and the darker the color, the more statistically relevant and pronounced the effect is, if I'm reading that correctly. I think I am. So looking at the bottom is, so on the y-axis, or the left side of that panel, is basically just the different categories of genes, and then on the bottom is, in the columns, is the animal that they got it from, that was treated with rapamycin relative to the control. So young female kidney, so D, you see young female's kidney, young female's liver, and then young male's kidney, young male's liver. And kind of the one thing that you kind of note, or at least I note immediately, is that there's a difference in kind of the patterns of between, there's a sexual dimorphism happening here in the expression profile of, to get all scientific-y, of male and female mice at the transcriptome level, right? Yes, Oren.
Yeah, I just wanted to note that here we see in D, we see in the ribosome.
Yeah.
So we see the opposite effect. But
exactly.
Also, ribosome is something that you would predict that rapamycin would reduce.
Yeah.
And now in males, you actually see an increase, which is weird.
Yeah. Yeah. So I don't know if that correlates to, as was mentioned before, maybe higher turnover of proteins, through autophagy. I'm not sure. Inflammation, it's interesting because pro-inflammatory responses are kind of reversed here. So on the left side, E, right? So let's see, I think that's females on the. Oh wait, no. Yeah, exactly. Females on the right and males on the left. Young, middle-aged, old, young, middle-aged, old. So inflammatory markers go up in females, and go down in males, which is, you kind of, and then, so you get all these interesting opposite effects, right? So in F, you have for metabolism, xenobiotic metabolism, cholesterol homeostasis, fatty acid metabolism. That goes up in females, goes down in males, both young and old. So there is this dichotomy, right? That's happening. That is a very pronounced effect that is linked. And these are heterogeneous. Both of these are from UHMET3, right? So it is the kind of the only thing that can really account for this is the sexual difference between the mice that I can think of, unless there's, again, another confounding variable, but that appears to be the case here. And somewhere in all of this data is something that might account for or maybe it's somewhere else. Somewhere in all of that data back there is something that might account for this difference in final lifespan between the male and female mice. Although, if you look at the maximal lifespan, the control males live less, right? So it looks like they're just getting as good as the females. So I didn't look at the average lifespan, but I'm just looking at the maximal lifespan. I'm just like putting a line right here. So it looks like, whatever is happening with the rapamycin is maxing out the lifespan of the male mice to that of the female mice. So whatever pathways are being activated in the male mice, it's having an opposite effect, but at the same time it's improving the lifespan of the male mice, but it's not having a negative effect on the female mice, which is good, right? So it's interesting to see. So xenobiotic metabolism, that's interesting because I don't know exactly what enzymes they're measuring for this, but xenobiotics are just anything you take in from the environment, like complex molecules. So breakdown of probably breakdown of rapamycin, right? So that goes up in females. So I don't know. Very simple question I have here. Are the levels of rapamycin in the mouse the same, or does it get broken down faster in female mice if you administer rapamycin? Because the metabolism is depressed here in xenobiotic metabolism and peroxisomes. I mean, I don't know. Just tossing that out there. I'm not sure if they mentioned that in the discussion, right? So anyway, just a thought. Does anybody have any other comments on this? Yeah, Warren.
Yeah, so I just hope that, because there are a lot of hypotheses here, right? Because you check a lot of stuff and they don't mention corrected p-value. I hope it is, but if it's not a corrected p-value, then I don't know. Maybe we shouldn't consider it very relevant, because there are many pathways, right? And they don't show you here all of the other pathways.
Mm-hmm. Yeah. So assuming that is the case, then there's a lot of hypotheses out there that you can toss out, and a lot of kind of controls. So one control is, it just occurred to me now looking at the pronounced metabolic differences, that is rapamycin hanging around as long as it is in females and males? Is it being excreted out faster? Is it Yeah. Anyway.
Yeah, but I meant multiple hypothesis testing with regard to p-values, right? So when you do 1,000 tests, you better correct for the. Yeah. So I don't know. It's a bit worrying.
All right. So that actually concludes, I believe, our mouse part of the paper. The Daphnia part is quite a bit shorter. I think this is the only first paper, actually not the only paper out there, but the first paper we've looked at with these critters here. They're actually pretty neat. That's pictures of them there. So really small, kind of like if you had as a kid sea monkeys, right? Those brine shrimp. About this, they're probably about the same size. The scale is 0.5 millimeters there. I think they're freshwater. I'm not sure. And these are the dosages: 10 nanomoles, 1 nanomolar, 0.1 nanomolar. So I have a comment to make here. But first of all, these are really, I remember reading about these critters. They have a pretty flexible phenotype. So I think they can really adjust their epigenome through environmental effects. Because I do remember reading somewhere that water temperature or predation can basically give them quite different, I don't know what you call that. So you can see the eggs here, this clutch of eggs. And they also have an interesting life cycle. They can go through sexual and asexual life cycles, so they can basically produce offspring parthenogenically, so without basically being fertilized. And a lot of other kind of cool things. They do note that, so this is major axis in millimeters. They look at, I think these are 3 different maybe types of water Daphnia, or maybe 3 different, can't be 3 different individuals. So these are Daphnia size measurements. So they tend to be, let's see, major axis, control D14. I think they're saying that's not significant at the lower level. So there you have a decrease in size of these Daphnia, and they look at survival probability. So they live about a little over 100 days, and basically all of these. So even the amounts of rapamycin that did not seem to have enough. Well, let's see. 0.1 nanomolar, not significant. D83, is that an individual or some kind of strain of water Daphnia? So everything down to 0.1 nanomolar had an effect, right, on, well, maybe not maximal, but certainly on the mean lifespan extension. One question I had when I read this, and I'm not sure, I don't think I saw it in, maybe it's in the material, in the method section, but so they come in males and females. So I'm just wondering, are these males or female water Daphnia that they're looking at here? Because we do know that the effect is definitely related to sex with mice, and I'm pretty certain that water Daphnia come in male and female varieties as well. And just looking at these pictures, this one here and this one here look like female water Daphnia. I don't know how a male water Daphnia would look like, other than it doesn't have eggs. But there are like, sexually dimorphic differences between them. So I'm not really sure, or this is just a mixed clutch of water Daphnia. So that would be like my question. If this sexual dimorphism effect of rapamycin also transcends species as well, because they do mention the size is also affected across species, and of course also the lifespan, of course. But is it a sexual effect in water Daphnia? And I don't know where let's see.
The method say we use females for all experiments.
Females for all experiments. Interesting. So my question now would be, Is it work for both males and females, or is the sexual effect flipped in water daphnia somehow? That would be weird. I don't know. And do male animals live longer or shorter? Controls in water daphnia. I don't know either. So yeah, any thoughts, comments on this?
Seems very. If you look down to panel C,
yeah,
the greatest increase in lifespan was from the tiniest dosage.
Yeah. Yeah. If that is a statistically significant difference, I'm not sure. Maybe it is. Like at this point here, right? Although it looks like the maximum is less. Although again, I don't know if that's a statistically significant difference. It's certainly the mean for all of them. If it looks like if you average it, it applies to all of them.
So there's a cool comment in the text associated with Figure 5. In the figures, it suggests they plot up these different concentrations, and at their lowest concentration, this 0.1 nanomolar, they say, well, the size doesn't actually change.
Right.
The text implies that the size, the growth rate still slows down at 0.1 nanomolar, because they say that their water fleas catch up.
Yeah, they caught up in size as they aged right here.
So I think that's really interesting, because if you go back to Figure 1 of the paper with the mouse intervention, there's two distinct effects that the rapamycin treatment has. It makes their mice smaller as adults, and it slows down their growth rate. And if you think that the size of the mice is like the main mediating variable here, it's the main thing the rapamycin does, either one of those could matter. It could be how quickly do you grow or how big do you get. Their water flea experiment suggests it's the rate at which you grow instead of how big you get too. And it's just one extra piece of data that I find that really interesting, because it suggests there's implications about how this is working that follow from that.
Yeah, because I mean, down here again, I don't see like the error bars, but you could see that it kind of looks like for the males the rate is slower, right?
Yeah.
And then of course it never reaches that point. It just stays that way. But could it somehow at a lower dose catch up, right? Even though it lags at some sort of early critical period of time. Yeah, interesting observation. Yeah.
Because there's also that paradox I mentioned earlier, where like really big species often live a long time. Whales live a long time. Well, they also grow for a really long time.
Their rate of growth is slower, yes.
So that would bring all of these observations together into a consistent model of what really matters here is the growth rate. And between the two experiments in this paper, it's consistent.
Yeah, and I'm going to toss something else out there. Growth rate, of course, is correlated, at least with mitotically active tissue with the rate of mitosis, right? So how quickly cells go through a cell cycle, I would assume. And cells, if they don't go into mitosis as quickly, might have more time during the G1 phase and S phase to repair any damage that might occur, which would suggest, and they mentioned that later in their discussion as one possibility. They said, well, maybe the somatic mutation load somehow is lowered in the early stages of the mouse, and what way could they possibly be, what mechanism could account for a lowering of the somatic mutation load than slowing down the cell cycle to allow any point mutations or any other things to potentially be repaired?
Yeah, maybe you're hanging out at the G1S checkpoint longer and fixing stuff. I mean, this isn't a scientific statement, but there's an aphorism. You can have it fast, you can have it quick, you can have it cheap. And one way to think about it is, with regards to mitosis, maybe you're trading off between fast and good, where good is like genomic fidelity of your daughter cells.
All right, I think we have the parts here, Michael, to design a paper and a study and test that. Using a simple model organism, right? Maybe not mice. Maybe we could stick to water daphnia or something even simpler, and just to see if we can demonstrate that using rapamycin and measuring somatic mutation load and correlating that with growth rate and ability to repair DNA, right? So that would make so other than like resetting the epigenetic clock early, yeah, I mean, it's kind of like being dosed with too much UV rays when you sunbathe early on in age, right? It's like if you don't accumulate all those somatic mutations, then you lower your probability of getting diseases of aging and other things, and that persists throughout life, right? So I'm not saying that's the whole story that's happening here, but it might be a partial or significantly partial story.
It's a very tempting story.
Yeah. Certainly not crazy. Yeah. All right, cool. I'm going to stop sharing here. Well, that was the paper, everybody. Raised a lot of interesting possibilities, and who knows, maybe we help tie up some of those loose ends at the end, to account for all of these differences. And why there's this, at least in mice, maybe that applies in water daphnia, I'm not sure too. In other words, why there's like this effect in between the two sexes of mice. That I'm not sure. It does, if you look at let's go. At least in this, it doesn't look like, it's hard to say if the growth rate was actually affected as much in the, it lags, but does it I think we need to look at more data points and see if there's how much of a difference there is. Maybe there's something to do with the cell cycle with the female mice, or maybe it's already maximized somehow, some other,
This is where I kind of wish they had a first derivative plot in Figure 1, because then you could directly compare the rates, male, female, drug control.
It's kind of interesting that the controls shrink a little bit at the end of their life, and the rapamycin mice don't shrink as much.
Let's see which. Are we taking a look at? Are we taking lifespan? Or are we taking a look at the weight? John?
We're looking at the weight, panels C and D.
Okay.
You see how the controls is orange, and they're going down in weight at the end of their life, but the rapamycins are not going down nearly as much.
Yeah, it's hard to say, right? Because this is like through the entire development to the mouse. So that's a good point, and
but it could be indicative of frailty and loss of muscle mass.
Yeah. I was about to say that could be another variable that's kind of overlaid, like the loss of mass here and the overall loss of mass here is for a different reason than the drop off of mass later in life, like in this window here, which, like you mentioned, John, could be due to frailty, loss of muscle mass and other issues that are aging related that are not as pronounced here because, there's less of a kind of frailty happening. Anyway. All right. Well, I actually have to leave fairly soon, but we have a few minutes that are open for discussion here. I like this paper because it, raised a lot of intriguing issues, and they did a lot of work here to try to account for these differences. And then they were upfront in the discussion to basically raise a lot of different possibilities as what can account for. So I think their next big step is lowering the dosage of rapamycin, and seeing these, what effects that would have on the mice as well. And I'm not sure if they mentioned in the paper if they went even lower doses with water daphnia, right? I don't know how, certainly keep going lower and lower and lower until you have a homeopathic solution. But I'm not sure how low they went, how low they could possibly go with pica moles and whatever. All right. Well, I'm just going to mention right now again that today is Giving Tuesday. So if you'd like to see more journal clubs and more conferences and more content and everything else that we do here at Lifespan.io, please give. Visit our website. All the info's there. And yeah.
Well, you'd hope so, seeing as I'm the person that puts it on there. So it is actually. We did it this morning. So you can you can find a link to the year-end fundraiser, and there's a bit of interaction this time round. We're trying something a little bit different. So there's 3 motifs, I guess you might want to call them, for next year, and we're gathering people's thoughts on which one of those it should be. So if people make a donation, for example, they can actually vote for the area of focus or motif that they prefer. It doesn't mean we won't be doing all of them, but obviously if there's a preferred winner, we're going to focus more on that. So it's a social experiment as well, so kind of gets people involved, doesn't it? So interesting. So I hope you've all enjoyed that about Daphnia and mice. It was a roller coaster of a paper, really. Oliver, it got really going when the Daphnia got into it. Who knew? So
yeah, one thing to mention about Daphnia is that you can really automate their measurements, right? Because they're little critters. You can have thousands and thousands of them, and you can have cameras with various systems tracking their motion and measuring their lifespan. So it might actually end up being an optimal organism to do various lifespan analyses for that reason, right? So it's a metazoan, has both sexual, asexual life cycles, and if a lot of the treatments that are tested recapitulate their effects in water Daphnia, then it might end up being a workhorse to basically screen for compounds.
Yeah, it's an interesting model.
I'm curious whether John Hemming or any of the others knows where people can get rapamycin.
Well, I get it from India. There are doctors who will prescribe it, and it is generally a prescription drug. And I get it from India. Couple of locations I got it from. My view is cycling is key, and I think the problem with a lot of these papers is they get the cycling wrong. And there's a lot to it. It's really interesting, but mine's from India.
Right.
And how often do you take it?
Well, I've taken it once topically and once orally. I have, because I do weekly blood tests, and I test a lot of. I'm one of these heavy biohacking people who does lots of weird things. I have a cycle of tests I intend to do, and I do intend taking rapamycin again. I see a logic for it, but I'm actually quite happy with my progress in lots of different ways, and it hasn't come up as yet. It probably will come up in December. We're not far from December.
Okay, well, hopefully you'll be joining us for our December Journal Club. And
I hope so.
Give us an update.
Yeah. Join you for December.
Right.
It's looking likely at the moment.
Great, fantastic. Well, I'll be seeing everybody then.
It's going to be a little bit earlier than usual as well, as a note, Oliver, because we close down on the Christmas Eve. The chances of motivating anybody to do any streaming during the Christmas holiday, pretty much zero. So we're probably going to have to go with like the third week, and let's maybe go with a bit of a belter of a paper. I've got a few ideas.
So that would be the 20th of December, I believe.
Yeah, we'll have a look at the dates and we'll announce it through the usual channels, of course. And
can I ask? It's not the same time as the investors event, because this was the investors, the Lifespan.io investors thing is now.
Yeah, I know. I get my notification. Is it always? Do we always?
Well, it was the last time. It was this time, and it's sort of. It would be nice if it was not, but there you go.
Okay.
I shall have a word with Javier then, who runs the LIN, because we've always had a Tuesday slot, for years. So I don't know why they moved it. But
they may have been on Tuesday, but it may be just a different cycling of Tuesdays.
I think it used to be a Wednesday. I think they used to do a Wednesday. I'll find out anyway. Yeah. I'll make a note of that, because if there's quite a few people that want to go to both, that's not really workable. So, right. Well, we'll have a think. We'll see what we can do. I mean, we're gonna solve aging. I'm sure we can solve clashes on a calendar, right? I'm sure we can.
Yeah.
Right.
So.
and on that note, we have the technology. So on that note, we will probably see you in a few weeks' time, just before Christmas.
Yeah.
If we can find a festive paper bonus for that, maybe we could have a look at cold therapy or something as a paper. Well, we like heat shock proteins. What about cold shock proteins and their application to longevity? There is some research, Oliver, so maybe we want to have a revisit of that.
Okay, so next month we'll be broadcasting from the inside of a sauna. All right.
And that's wrong proteins, mate. But yeah, I mean, a sauna works for me, or a hot tub.
Okay. All right, folks. Well, thanks for attending. It was a fun paper, and as always, I really enjoy everybody's discussions and input on this. It makes it fun. It makes it a real journal club.